Welcome to my poster presentation. My project is based on the discovery of snoRNAs which regulate the expression of their host gene. So first of all, what is a small nucleolar RNA or snoRNA? It's a highly structured and highly abundant non-coding RNA, ranging in size from 60 to 300 nucleotides. snoRNAs are divided in two groups, box CD snoRNA and box HACA snoRNA. The canonical functions of box CD snoRNA and box HACA snoRNA is, respectively, to guide the 2' O methylation and the pseudoresylation of ribosomal RNAs and small nuclear RNAs. There is 1,541 snoRNAs currently included in our annotation, from which approximately 38% are expressed according to our RNA-seq analysis from 7 human healthy tissues. And from these expressed snoRNAs, more than 86% are embedded in other genes, mostly in introns. Here's a general overview of how snoRNAs are produced. A host gene containing the snoRNA sequence is transcribed and spliced into a mature mRNA and lariats. In this case, a lariat containing the snoRNA and its bound snoRNP core proteins. Lariat will be debranched and degraded by 3' and 5' exonucleases, and the snoRNP core proteins bound to the snoRNA sequence will prevent subsequent degradation, leading to mature snoRNP. In the last 2 decades, many other non-canonical functions have been attributed to snoRNA, such as alternative splicing, 3' end processing, exosome recruitment, chromatin remodeling, and protein trapping. And even though snoRNAs have been shown to be involved in a lot of cellular processes, only a handful of snoRNAs have been shown to have non-canonical functions. Our research hypothesis is that the non-canonical functions of snoRNAs are much broader than what we currently know. To investigate my hypothesis, I used data from 3 groups who developed different but similar techniques that aim to map the RNA-RNA interaction in the cells. These methods are called Paris, LIGR-Seq, and SPLASH, and the studies were all released in 2016. These methods are based on the use of the psoralen derivative, which produces a reversible crosslink between 2 paired RNA molecules. This psoralen derivative is added to the cells, which are then irradiated to induce a crosslink. A nuclease step is used to concentrate the interacting region, and subsequent steps of proximity ligation and decrosslink produces a single RNA molecule that is used to do a library preparation. RNA-seq is then performed to retrieve the RNA-RNA interaction that were present in the cells. From our reanalysis, we found that more than 25% of all the detected snoRNAs showed at least one interaction with their host gene. And from those, more than half displayed an interaction in the same intron where the snoRNA is located. Here's one of the most interesting interactions that we found, which is between the SNOR2 snoRNA and the downstream intron region of its host gene EIF482. This interaction was seen in 3 different datasets, 1 from Paris and 2 from LIGR-Seq. One of the key features that makes this candidate interesting is the fact that the next exon downstream of SNOR2 is a cassette exon. Using an RNA duplex prediction tool called intronic, we observed a strong predicted interaction region within SNOR2 EIF482 interaction, as represented here by the purple and the pink color. In order to get more insight into this interaction, we use the RNA secondary structure prediction tool mFold to fold SNOR2 with the downstream intron region. Here's the predicted secondary structure of SNOR2, which is a pretty standard conformation for a box CD snoRNA. Now this is the predicted folding of SNOR2 with the downstream intron region in gray. This structure has a low minimum free energy, suggesting that this conformation is favorable. In this intron, the predicted branch point, as represented here by the red star region, is located in the middle of the predicted RNA interaction region, which suggests that this base pairing of the branch point with SNOR2 sequence could impact the splicing of the next exon. Next, we investigated the possibility of a stable SNOR2 intron intermediate using our and other RNA-seq datasets from 7 human healthy tissues, 6 cancer cell lines, and 2 reference RNA. Surprisingly, we were able to see coverage in the downstream intron of SNOR2 for some datasets, as represented by the red arrows, with generally higher coverage in cancer cell lines. Then, we wanted to know if snoR2 intron was somewhat linked to the splicing of exon 4. To answer this question, I used the psi value, which in this case measures the level of transcript containing exon 4 over all EIF42 transcript, and made a correlation of this value with the coverage of the intron region located downstream of snoR2. As you can see, there is a strong, significant negative correlation between the psi value and the intron coverage. In conclusion, when there is no observable coverage in the intron downstream of snoR2, transcripts having exon 4 are mostly generated, leading to the production of the protein. In contrast, when a higher coverage of the intron is observed, a higher amount of transcript lacking exon 4 are produced, leading to transcript that will be targeted and degraded by the NMD and possibly less protein generated. In contrast, we found no correlation between the psi value of exon 4 and snoR2 alone, confirming that snoR2 intron RNA, but not the mature snoR2, is linked to the splicing of its host gene. Based on our results, we built a model of EIF42 regulation by the embedded snoRNA snoR2. There's a balance between the folding of snoR2 with itself and the folding with the downstream intron. When snoR2 folds in a canonical way, this leads to the production of transcript having exon 4, which will eventually give EIF42 protein and mature snoR2 snoRNP. In contrast, when snoR2 sequence folds with downstream intron, this will lead to the production of a stable RNA intermediate snoR2 intron, as well as a transcript lacking exon 4, which will subsequently be degraded by the NMD pathway. I would like to thank my lab and our collaborators on this project, as well as our funding agencies. Thank you for stopping by.